The Bcl-2 family of proteins are critical regulators of programmed cell death, or apoptosis. These proteins are characterized by the presence of up to 4 conserved Bcl-2 homology (BH) motifs. This slide provides an overview of the structural biology of the Bcl-2 family, their complex interactions, and their emerging role as targets for drug discovery. The 3D molecular structure shown highlights the alpha helices of the Bcl-2 proteins in different colors against a dramatic, dark background. This highly detailed molecular rendering helps visualize the complex structures and interactions of these important proteins. I'll dive deeper into the structural features, binding interactions, and drug discovery applications in the next few slides. The Bcl-2 protein family has a long evolutionary history, dating back to early multicellular organisms like sponges, anemones, and hydra. Over time, these proteins diverged into pro-survival and pro-apoptotic members, creating a complex network that regulates cell death processes. Interestingly, viruses have incorporated Bcl-2 proteins into their own genomes, demonstrating the evolutionary importance of these proteins in evading host immune responses. This evolutionary timeline and divergence of the Bcl-2 family highlights their critical role in the regulation of cell fate and survival, from the earliest multicellular life to modern-day organisms and viruses. The Bcl-2 family of proteins play a crucial role in regulating apoptosis, or programmed cell death. This slide outlines the 3 main subgroups within the Bcl-2 family: pro-survival proteins, pro-apoptotic proteins, and BH3-only proteins. Pro-survival proteins like Bcl-2 and Bcl-XL contain multiple Bcl-2 homology (BH) motifs and help prevent apoptosis. Pro-apoptotic proteins like Bax and BAC also have multiple BH motifs, but they trigger apoptosis. BH3-only proteins like BIM and PUMA only have the BH3 motif, but they can activate the pro-apoptotic proteins to induce cell death. Understanding the distinct roles of these 3 subgroups is key to comprehending the complex regulation of apoptosis. The Bcl-2 family of proteins play a crucial role in regulating apoptosis, or programmed cell death. These proteins contain conserved sequence motifs known as BH, Bcl-2 homology motifs, which are important for their structure and function. The BH1 and BH2 motifs form a critical binding groove that interacts with the BH3 domains of pro-apoptotic Bcl-2 family members. The BH3 motif is the minimal requirement for apoptosis-inducing proteins, containing a signature sequence that is crucial for binding. The BH4 motif is the most weakly defined of the BH motifs, but is found in all multi-motif Bcl-2 family proteins. Understanding the roles of these different BH motifs is key to elucidating the complex mechanisms of apoptosis regulation by the Bcl-2 family. The Bcl-2 family of proteins share a highly conserved structural architecture, despite differences in their amino acid sequences. This central hydrophobic helix, alpha 5, is surrounded by up to 8 other helices, forming a distinctive fold. This fold is remarkably preserved across the entire Bcl-2 family, highlighting its evolutionary importance. Pro-survival Bcl-2 proteins like Bcl-XL have a hydrophobic groove formed by the BH1, BH2, and BH3 motifs, which serves as the binding site for BH3-only proteins. In contrast, pro-apoptotic Bcl-2 proteins like Bax undergo conformational changes during activation that expose their BH3 domain and facilitate oligomerization. Even viral Bcl-2 homologs maintain this core fold, despite having minimal sequence identity to their cellular counterparts. This structural conservation across the Bcl-2 family underscores the critical functional role of this distinctive protein architecture. The Bcl-2 family of proteins play a crucial role in regulating cell death and survival. A key feature of these proteins is the C-terminal transmembrane (TM) region, which acts as a membrane anchor. This TM region forms a helix that can interact with the BH3 binding groove of the protein, influencing its function. The TM region is not just a passive anchor, but actively participates in the protein's activities, like forming pores in membranes and interacting with specific lipids. Deleting this TM region, while leaving the BH3 binding groove intact, can impact the overall functionality of the Bcl-2 proteins. Understanding the structure and role of the TM region is crucial for understanding the biology and potential therapeutic targeting of these important cell death regulators. BH3-only proteins are the sentinels that detect cellular stress and initiate the apoptotic cascade. They act as the initial triggers, integrating various apoptotic stimuli. With the exception of BID, BH3-only proteins are intrinsically disordered, giving them flexibility to interact with multiple partners. Each BH3-only protein has a unique binding profile, selectively antagonizing specific pro-survival Bcl-2 proteins. Some BH3-only proteins can directly activate Bcl and BAC, leading to their oligomerization and pore formation, a critical step in apoptosis. The intrinsic disorder of BH3-only proteins provides them with conformational flexibility, allowing them to interact with multiple binding partners. These IDPs are tightly regulated and can be degraded at the proteasome without post-translational modification, providing another layer of control. The presence of linear motifs in IDPs enables splice variation to modify their behavior by combining or deleting interaction sites, refining their functional diversity and regulating biological activity. BH3-only proteins like PUMA bind their targets through an induced fit mechanism rather than conformational selection, as demonstrated by proline point mutant studies. The key difference in apoptosis mechanisms between C. elegans and mammals is the role of the mitochondria. In C. elegans, the process is more direct. The Bcl-2 ortholog CED9 binds the adapter CED4, which then activates the caspase CED3 to trigger cell death. In mammals, the Bcl-2 family has many more members, and they regulate cell death through mitochondrial outer membrane permeabilization, MOMP. This MOMP releases apoptogenic factors like cytochrome C, which then initiate a cascade leading to caspase activation and cell death. So the worm system is more streamlined, while the mammalian process involves more complex regulation at the mitochondrial level. MOMP is a key step in the apoptosis pathway, where mitochondrial outer membrane permeabilization allows the release of cytochrome C and other death factors. The process begins with activation of BH3-only proteins, which neutralize pro-survival proteins or directly activate Bcl and BAC. Activated Bcl and BAC then undergo conformational changes that expose their BH3 domains. Bcl and BAC then oligomerize and insert into the mitochondrial outer membrane, forming pores. These pores allow the escape of cytochrome C and other death factors, triggering the apoptosis cascade. While the general mechanism is understood, the exact molecular details of how Bcl and BAC breach the membrane and the precise structure of the pores remain uncertain. Further research is needed to fully elucidate this critical step in programmed cell death. Viruses have evolved to incorporate Bcl-2 proteins into their genomes as a way to evade the host's immune defenses. These viral Bcl-2 homologs, while having little or no sequence similarity to mammalian Bcl-2 proteins, still maintain the characteristic Bcl-2 fold and functionality. Examples of these viral Bcl-2 homologs include BHRF1 from Epstein-Barr virus, M11L from myxomavirus, F1L from Vaccinia virus, and N1L also from Vaccinia virus. The ability of viruses to co-opt this important apoptosis-regulating protein family highlights their evolutionary adaptability in the ongoing arms race between hosts and pathogens. Viral Bcl-2 proteins like BHRF1 and M11L are able to inhibit apoptosis, or programmed cell death, by binding and sequestering pro-apoptotic Bcl-2 family members. This prevents cell death and allows the virus to replicate within the host cell. Some viral Bcl-2 proteins, such as murine gamma herpesvirus 68M11, KSHVK's Bcl-2, and human adenovirus E1B19K, can also target the Bcl-1 protein to inhibit autophagy, a cellular process that can lead to cell death. Interestingly, poxvirus proteins like A52, B14, and N1 are able to engage the NF-kappa B signaling pathway, demonstrating that the Bcl-2 fold can have functions beyond just regulating apoptosis. The versatility of viral Bcl-2 proteins is further illustrated by F1L from Vaccinia virus, which can inhibit both caspase-9 and the NLRP1 inflammasome, allowing the virus to evade host immune responses. Viral Bcl-2 proteins have evolved unique structural features while maintaining their core functionality. F1L forms domain swap dimers, giving it two binding grooves. A52, B14, and N1 dimerize through an alternative alpha-1 alpha-6 helices interface. N1 has separate binding sites for apoptosis inhibition and NF-kappa B inhibition. BHRF1 has an extra alpha-1 foot helix in the alpha-1 alpha-2 loop position, an additional structural element. Bcl-2 proteins play a critical role in regulating apoptosis, or programmed cell death. Dysregulation of Bcl-2 proteins is a hallmark of many diseases, particularly cancer. The Bcl-2 family includes both pro-survival and pro-apoptotic members. Mechanisms of Bcl-2 dysregulation include overexpression of pro-survival proteins, mutations in pro-apoptotic proteins, and altered regulation. These changes can allow tumor cells to evade cell death and contribute to malignant transformation. The pie chart shows the breakdown of Bcl-2-related diseases, with cancer being the most common at 65% of cases. Bcl-2 was first discovered in the 1980s due to its role in non-Hodgkin's lymphoma. It was cloned and characterized in 1986, kickstarting research into the Bcl-2 family. In 1988, the critical role of Bcl-2 in promoting cell survival was demonstrated, establishing it as an inhibitor of programmed cell death. In the mid-1990s, the structure of Bcl-XL, another Bcl-2 family member, was described in seminal papers. Over the past 15-plus years, the development of BH3 mimetic drugs targeting Bcl-2 family proteins has led to cancer therapies reaching clinical trials and FDA approval. The overexpression of pro-survival Bcl-2 proteins is a common feature in many types of cancer, making them attractive targets for therapy. By neutralizing these Bcl-2 proteins, we can restore the normal apoptotic process in cancer cells, leading to their death. Small molecule BH3 mimetics can bind to the groove of pro-survival Bcl-2 proteins, antagonizing their action and removing the blockade on back-induced apoptosis. Understanding the molecular structures and interactions of Bcl-2 complexes has been crucial for structure-guided drug design, allowing us to develop highly specific inhibitors. Several BH3 mimetic drugs have progressed to clinical trials, and some have even received FDA approval for treating specific cancers, demonstrating the success of this targeted approach. BH3 mimetic drugs are a major success story in structure-based drug design, They target the hydrophobic binding groove and pocket that interacts with the conserved BH3 motif. The first BH3 mimetic, ABT-737, was discovered in 2005 using NMR-based screening. Venetoclax, an orally bioavailable version, was the first FDA-approved BH3 mimetic in 2016 for chronic lymphocytic leukemia. BH3 mimetics are estimated to have a $3.5 billion market value by 2025. They can achieve response rates up to 70% in certain blood cancers with high Bcl-2 expression. These drugs represent an important new class of targeted cancer therapies. APT-737 was the first BH3 mimetic drug, binding to Bcl-2, Bcl-XL, and Bcl-W with high affinity. It was discovered through structure-activity relationships and medicinal chemistry. APT-263, also known as navitoclax, is an orally available version of APT-737 that targets the same Bcl-2 family proteins. However, its inhibition of Bcl-XL can cause thrombocytopenia, or reduced platelet count, as a side effect. APT-199, or venetoclax, is a more selective Bcl-2 inhibitor that avoids targeting Bcl-XL, thereby avoiding the thrombocytopenia issue. It has shown strong clinical success and received FDA approval for certain leukemias. Newer compounds are emerging that target MCL1, another anti-apoptotic Bcl-2 family protein. These include constrained peptides based on the MCL1 BH3 motif, as well as small molecule inhibitors discovered through fragment-based approaches. Targeting the Bcl-2 family proteins is a key approach in cancer therapy, but achieving specificity has been challenging. While inhibiting close relatives like Bcl-XL, Bcl-2, and Bcl-W has been successful, selectively targeting other pro-survival proteins like MCL1, A1, and Bcl-B has proven more difficult. This is important because these other proteins can provide alternative survival paths for tumor cells. Broad-spectrum inhibition can also lead to side effects, like the thrombocytopenia, reduced platelet count, caused by APT-737 due to its impact on Bcl-XL. The development of APT-199, venetoclax, which selectively targets Bcl-2 without affecting Bcl-XL, demonstrates how improved specificity can reduce side effects while maintaining therapeutic efficacy. This selective approach has been successful in clinical settings and represents the future direction for BH3 mimetic drug development. Viruses are responsible for about 12% of all human cancers, with Epstein-Barr virus, EBV, alone causing around 200,000 cancer cases worldwide annually. EBV-BHRF1 is a viral Bcl-2 protein that is constitutively expressed at low levels in EBV-transformed cell lines and confers chemoresistance in Burkitt's lymphoma. This makes EBV-BHRF1 a promising target for therapeutic intervention. A potent protein-based inhibitor of BHRF1, called Bindi, has been developed with picomolar affinity and substantial selectivity over endogenous Bcl-2 members. Small molecule antagonists have also been developed against Vaccinia virus N1, and peptide inhibitors against murine gamma herpesvirus 68M11, showing the potential for targeting viral Bcl-2 proteins. Viruses have evolved their own versions of the Bcl-2 protein, which is a key regulator of apoptosis, programmed cell death. These viral Bcl-2 homologs help the viruses evade the host cell death response and promote their own survival and replication. The table summarizes some key viral Bcl-2 proteins, the viruses they come from, and their important functional features. For example, the Epstein-Barr virus BHRF1 protein requires neutralization of both the BIM and BAC proteins to inhibit apoptosis. The Kaposi sarcoma virus K's Bcl-2 protein can inhibit apoptosis without directly binding to the host BACs and BAC proteins. Understanding how these viral Bcl-2 proteins work provides insights into the complex interplay between viruses and their host cells' death pathways. The central question we'll be exploring today is how the Bcl-2 family of proteins regulate apoptosis, or programmed cell death, at the molecular level. One key unresolved issue is the mechanism of Bax and Bac activation by BH3-only proteins. We still don't fully understand the conformational changes that occur during this process. Another open question is the exact mechanism by which Bax and Bac breach the mitochondrial outer membrane to induce apoptosis. The structure of the pores they form is also not well characterized. We also need to better understand the molecular basis for the interactions between pro-survival and pro-apoptotic Bcl-2 proteins, both in the cytosol and at membranes. Finally, we want to explore how the activities of Bcl-2 proteins are regulated in different cellular contexts, and what role post-translational modifications play in this regulation. The Bcl-2 family of proteins play a crucial role in regulating apoptosis, or programmed cell death, across different organisms. In mammals, we see the most complex network, with around 20 Bcl-2 family members that form an intricate regulatory system. This includes pro-survival proteins like Bcl-2, Bcl-XL, and MCL1, as well as pro-apoptotic proteins like Bax and Bac, and BH3-only proteins that act as sensors and initiators of apoptosis. In contrast, simpler organisms like the nematode C. elegans have a more streamlined apoptosis pathway, with just one Bcl-2 ortholog, one adapter protein, and one caspase. Drosophila, the fruit fly, contains Bcl-2-like proteins but lacks the BH3-only proteins found in mammals, suggesting a different apoptosis mechanism. Even more primitive multicellular organisms like hydra and sponges contain multiple Bcl-2-like proteins, indicating that apoptosis regulation evolved early in metazoan history. The variations in Bcl-2 protein networks across these organisms provide insights into the evolution and complexity of apoptosis regulation. Bcl-2 family proteins have important functions beyond just regulating apoptosis. They play a key role in calcium homeostasis, influencing cellular signaling. Bcl-2 and Bcl-XL can reduce calcium release from the endoplasmic reticulum. Bax and Bac promote increased calcium release. Bcl-2 family members also participate in mitochondrial fusion and fission processes. This affects mitochondrial morphology and function, impacting cellular metabolism and energy production. Pro-survival Bcl-2 proteins can bind to Bcl-1, a key regulator of autophagy. This inhibits autophagosome formation, creating a complex interplay between apoptosis and autophagy pathways. Bcl-2 family proteins are regulated by a variety of post-translational modifications. Phosphorylation is the most common, with over 40 known phosphorylation sites across the Bcl-2 proteins. Phosphorylation can enhance or inhibit the anti-apoptotic functions of Bcl-2 proteins. Ubiquitination targets Bcl-2 proteins for proteasomal degradation, regulating their cellular levels. Other modifications like acetylation and methylation can alter protein interactions and localization. This additional layer of regulation allows fine-tuning of Bcl-2 protein activity and function. Bcl-2 proteins play a critical role in regulating cell death, and their interaction with membranes is key to their function. Membrane targeting is essential for Bcl-2 proteins to carry out their regulatory activities. The C-terminal transmembrane (TM) region of these proteins inserts into the mitochondrial membrane. Lipid interactions, particularly with cardiolipin, help localize Bcl-2 proteins to the mitochondria. The membrane environment can induce conformational changes in Bcl-2 proteins, altering their structure and function. In the case of Bax, These structural changes allow the protein to oligomerize and form pores in the mitochondrial membrane, facilitating the release of cytochrome C and triggering apoptosis. The slide provides an overview of the key structural techniques used in Bcl-2 protein research. X-ray crystallography has been instrumental in determining the high-resolution structures of Bcl-2 family proteins, including Bcl-XL, as well as numerous protein-ligand complexes. Nuclear magnetic resonance, NMR, has been used to study the structure and dynamics of proteins like Bcl, and to investigate protein-ligand interactions, including the SAR by NMR approach that led to the development of the inhibitor Apt737. Cryo-electron microscopy is an emerging technique that has the potential to reveal how Bcl oligomers form pores, by studying larger complexes and membrane-associated structures. Computational methods, such as molecular dynamics simulations and structure-based drug design, have been crucial for understanding protein dynamics and developing inhibitors targeting the Bcl-2 family. The future of Bcl-2 research holds exciting possibilities, from determining the structures of full-length Bcl-2 proteins in membrane environments and oligomeric complexes, to creating selective inhibitors for previously untargeted Bcl-2 family members. Exploring combination therapies that leverage the synergistic effects of BH3 mimetics with other cancer treatments could lead to more effective treatment strategies. Developing therapeutics that target viral Bcl-2 proteins could open up new avenues for treating virus-associated diseases. These advancements in structural biology, drug development, combination therapies, and viral targets will drive the field of Bcl-2 research forward, paving the way for innovative solutions to complex health challenges. The clinical translation of Bcl-2 research has been a major success in targeted cancer therapy. Venetoclax, a Bcl-2 inhibitor, has shown remarkable efficacy in treating chronic lymphocytic leukemia and acute myeloid leukemia. There are currently 15+ active clinical trials testing BH3 mimetics, which target Bcl-2 family proteins, in various cancer types. 3 Bcl-2 targeting drugs have been approved by the FDA for cancer treatment. Venetoclax has shown a 70% response rate in certain blood cancers. Bcl-2 inhibitors are showing promising results in 8 different cancer types. Ongoing trials are exploring combination therapies, expanded indications, and novel compounds with improved specificity. Targeting protein-protein interactions, like those involving Bcl-2 family proteins, has proven to be a successful approach in cancer treatment. The Bcl-2 family of proteins are key regulators of cell fate, acting as guardians that balance life and death signals. Despite differences in their amino acid sequences, Bcl-2 family members share a highly conserved 3D structure in alpha helical bundle that provides a binding groove for their BH3-bearing antagonists. This complex network of protein-protein interactions between Bcl-2 family members is critical for regulating mitochondrial outer membrane permeability and the initiation of apoptosis. Dysregulation of the Bcl-2 family is implicated in many diseases, particularly cancer, making these proteins important therapeutic targets. The structure-based design of BH3 mimetic drugs that target Bcl-2 proteins has led to clinically approved cancer treatments, demonstrating the power of structural biology in drug discovery.